All right, so today I'll try to walk you through, and I have, how much time? Oh, never mind. All right, I'll walk you through our approach to machine learning for drug discovery at GCS, which is a new org formed to encompass lots of different, as John mentioned this morning, lots of different early to phase 2 research activities at Genentech and at Roche. And so this is a GRED, PRED organization. Okay, I didn't put that there, but thanks, yeah, a little advertisement. Okay, all right. And so before I start, I'll just talk about how we organize the unit that's putting together this inner loop of drug discovery. And so we'll get to how this fits into the overall process of drug discovery at Genentech. But for us, one of the first things we had to think through was what's the right team structure for approaching using generative AI, but also approaching the process of going into the drug modalities and integrating into the experimental lab workflows, winning over the head of antibody engineering, winning over chemists who are doing things in an industry that for all of the difficulty is still fundamentally profitable and does make life better for a substantial number of patients, even if you do it the old way. So you still have to, you have a certain number of folks on your team that have to be really deeply embedded in the domains so that we can actually speak the same language and get these machineries put into the lab in such a way that they're connected to not just the robots, but also the people and the decision makers across the org. And so we actually have two somewhat old-fashioned divisions, which is for each of the main modalities, we have a subdivision that's just devoted entirely to that drug discovery modality. You can imagine, and if you did imagine so, you would be correct, that lots of other modalities get slotted into those. So peptides go into, who thinks peptides go into small molecules? Who thinks they go into large molecules? All right, we put them in small molecules because we're sick of some of the informatics associated with peptides because we're very lazy. Large molecules, we put in nucleic acids and other things like that. But generally speaking, this lets us have two hyper-focused columns that are thinking about where the rubber meets the road, how to make the molecules. We also have our astronauts in Frontiers. This is a group led by Kyung-Young Cho, and there we're focused on open access, accessible, publishable machine learning research that crosses lots of different subject areas at Roche that include things that are beyond molecular discovery, like causal inference, active learning, I'll talk more about that later, and other things like that. So the Frontiers Research Group is, in many ways, our skunk works. It's where we go when we're confused. It's where we go when we have opportunities that go beyond just drug discovery. We also have our ML engineering group. We thought that it was very important to make sure that we had a group that's very tuned into the ethos of this conference. These are our folks who liaise with AWS and with other vendors when we're thinking about computation and we're thinking about computational technologies. They're also the folks who build our core internal system for Lab in the Loop. And then lastly, we have, well, this is supposed to say LMM, like large language models. I'm not sure what happened there. But we also have a large language model and agentic framework group. And that's the group who's building out the last part of this talk today, which is our systems for running the workflows that we prototype in the small molecule and large molecule columns, but running them in a way that's accessible and deployable across the entire Roche family with an agentic front end. All right, and so, you know, in the end, I think there are lots of different things that you have to think through to get this to work. I remember in the early days of learning about chemistry, you learn about rate-limiting steps. And I think when I first learned about rate-limiting steps, I thought that can't be right. You know, there can't be like 100 steps in this biochemical pathway and the only one matters is the slowest one. That doesn't seem fair. You know, and it turns out that that is how you think about process optimization and operations research. You have to focus on rates and bottlenecks and bandwidths that span log units. And so in that sort of mindset of really making sure we figure out how machine learning powers drug discovery from end to end, it really emphasizes certain topics more than others. And I think one of the most important topics that it emphasizes is active learning. And so I don't have a pointer, but it's Lab in the Loop active learning This has been something that we've put a lot of research effort into, both at Roche and Genentech, and this is a big part of what the Frontiers group focuses on. And so we'll get into this a little bit more with some geometric descriptions and some numbers, some parameters, so we can make this tangible. But if you have powerful generative AI, it can typically produce billions of hypotheses, of which you can maybe come up with high throughput methods to test millions. And shift the log units appropriately, depending on what we're talking about, because there's definitely places in Genentech where we test 10 to the 14 things at once, right? Library technologies, things like that. But usually you can think up or generate many log units beyond what you can test. And when you test, you also have tiers of high throughput tests versus more detailed parameters. Immunization, high concentration behavior. There's many things that we care about with antibody drugs. There's many things we care about with small molecule drugs that aren't really amenable to high throughput. And so how do you figure out ways of designing and driving both your laboratory, your slow lab, and your automated lab so that they stay in sync and produce the best hypotheses and ultimately the best drugs and ultimately the best medicines? So if you're interested in the real beating heart of Lab in the Loop, send me a note and I'll forward you a handful of our favorite things that we've been able to release on the active learning front. And most of what we've produced in this space of active learning we've been able to publish. All right, we of course need deep predictive power, generative AI that can design molecules, that can predict structures, that can think things up. This is what's dominated most of the media narrative around what AI can do for drug discovery. And I get it, it's very exciting. And we do have efforts in de novo hit discovery and structure prediction and modeling. And I see a few old faces like John in the audience who are from 20 years of struggling with these questions. And so we're very excited to see as much progress as we have. And we certainly have those efforts at Genentech and across the industry. Agents and orchestration, I'll get to that at the end with some proper examples. And I think we saw some furiously exciting examples this morning of what agents can do and how some of what we do in research, some of what we do in commercial, some of these things can use standardized tech stacks, things that are maintained the same way, things that are developed the same way. And so I think there's also this really amazing process integration aspect to that. I think cross-modality by design, this is me just saying we're at the point where I have a small molecule group and a large molecule group, but I shouldn't. We should just think atoms are atoms, interfaces are the key. There's different ways of building function. Think about them geometrically. Think of them by squared angstrom surface area. Don't think of them how you make the molecule. We're not totally there yet, but that's one of our guiding lights. And then the last thing to say is just that everything we're doing here today, everything we're talking about here today is also a great motivator for just going and process optimizing big parts of how we put things together across the organization. And that has been really exciting. And I've been very lucky to be at Genentech, which is an innovator in high throughput biology, molecular engineering, chemistry, and many other things. And so between Genentech and our partners, we've had a lot of fun using what we do with agents and LLMs and generative AI to just go back and look at where are the rate limiting steps in the physical lab space. And that's been a lot of fun. The thing where I can sort of point you to papers and other bits and pieces so you can see the details, you can see the numbers, and you can see what fraction increase in rates and various things is in the large molecule space. This is something where we've been going for 4 years. The other modalities are a handful of years behind this. And this was the modality at Genentech that we used to really kick the tires and get Lab in the Loop working and integrate it across essentially the entire large molecule pipeline. And so at this point, this loop is integrated in the majority of projects and certainly in all the most exciting active projects right now in antibody engineering. This is also something that's deployed at Roche and Dia and other places like that. But the idea here is we start off seeding these models with the data at the company from prior projects, from the initial experiments that people typically do at the beginning of any antibody engineering campaign, and public data sources. We build a model, but the key thing is that once we build that model, we test it using active learning frameworks. So we figure out examples and predictions to test, designs to test that have a chance of being right, but also have some way in which they optimally improve the model. So you want things that look great, but have a lot of error. You want things that are different from other things. You want things that expand the space that you've explored. In a high-dimensional space, like a small molecule or a sequence, it is unthinkable how to do that. I employ people who are smarter than me, like Natasha Tavasovska and Jiwon Park, who are two of our key scientists in Frontiers. And they've built out things that don't worry about the sheer dimensionality here. And I can't say that I fully understand everything they've done. That model picks things out. We build them out. We make them. We physically embody them. We test them in the laboratory. We allow people to kind of tinker with them because every once in a while, something that the model likes is implausible for all sorts of simple reasons, whether chemistry or biophysical. And then we turn this cycle multiple times. And this is something that has been an active contributor to many projects, including projects that have navigated key transition points. I would like to just quickly have a shout out to Sarah Mustafavi, who's our new Vice President of Computational Biology and Translation. Ultimately, what we want to do at Genentech and what we are doing now is we want to take our advanced systems for prioritizing targets, which again, use all the technologies that we're all talking about today, and our advanced lab-in-the-loop systems, and have a system where we start to think of target families, target and pathway families that interact with multiple compound families. As we're developing molecular hypotheses, we're thinking of compounds and targets together as tuples, as data structures that are significantly more advanced than what we've typically thought through. All of that powered by a data backend built by Ryan Copping and the Catalyst team at Genentech. This is a system that is beginning to be in operation. I'm talking about these two cyan boxes at the bottom, which are our agentic and LLM framework for orchestration, and our lab in the loop. But Sarah could give an equally exciting talk about how we select our targets in the first place. And again, being at a place that is such an innovator in genomics and high throughput biology, it makes this very exciting for both of us. But again, we're going to go back to an example where I can show you quite a bit of real embodied examples, ones that we have permission to release, and so we can all dig into the details offline. But again, this is the punchline that I guess stole from myself by kind of just saying all this in the intro. I could, I guess, end the talk now. But we have some number of things. And we'll just say that we have many orders of magnitude more than we can test. These are things that could be generated by unsupervised methods. So show me compounds that are likely synthesizable. Ultimately, we want, we want this to be guided by oracles. So one of the key things that was the founding principle of what we're doing with Lab in the Loop at Genentech is that we have unsupervised methods that can learn how to produce structure, produce sequence, produce small molecules. We slowly work to join those molecules into more unified universal molecular encoders and decoders. We guide those with oracles that know about drug-like properties. But they can still generate vastly more than we can test, so there's an inference step where we start to think of not just the individual predictions, but how they relate to each other in the space of all these properties. We select things that are likely to improve the model and also have a diverse set of properties. And this is important, especially when you get into problems late in the stages of drug discovery, to have a diverse ensemble. Ultimately, for the more detailed tests, the more expensive tests, the things that involve animals or high concentrations or large quantities of material, you very rapidly get down to being able to only test 100 things if you want to move fast. Usually it's more about speed than cost. Let's go through this just in a very overly simplistic way. If we just think of two properties, and here I'm showing real data for yield in a small plate-based format. So a small number of cells, cheap constructs from the cheapest way to get the DNA from Twist, the cheapest way to do everything. Lots could go wrong. Very little of it's process optimized, and so the only way you process optimize this is through the machine learning, through the construct. So that's the expression yield, and that's the affinity in PK, or PKD, sorry. All right, so the idea is this classic idea, which is that if you have two properties, you want to pick points that increase the volume of the points you've seen. So you count the origin, and then you draw a box around this in a Manhattan distance style, and you push out the volume as much as possible. Anytime you hear someone say Pareto front, that's what they mean. It's just a very fancy way of saying explore new space, break new ground, look at combinations you haven't seen before. And in a multidimensional space, you get a lot of volume for combinations you haven't seen. It all comes out in the wash if you just optimize volume. But the reason why Natasha and Jiwan are geniuses is because they figured out how to address this problem. And this is the problem that hurts us in the real world, which is that properties aren't just this Pareto front thing, this volume thing, it works when the properties are measured for everything and they're not so correlated with each other. Now, if they're correlated with each other and they have structure that way, well, you can solve that mathematically. You can figure that out with projection methods and all kinds of great things. You basically project those properties into some new sort of principal component-like property space. That's a problem we've solved since the 70s. But the other problem that's a little more difficult is that if my antibody doesn't express, Yan Wu will not do anything else with it because you can't make it. If it doesn't bind, she's also a pretty fast-moving character, so she's not going to say, oh, I really care about your science. I'm going to do a whole bunch of other expensive experiments with something that doesn't even bind the target. And the list goes on and on. If we see a liability, we stop. These, of course, this is the very top of this tree, but this bush goes down a few layers and is definitely a burning bush. Because what it means is I've got data for everything here, half the things here, less than a third of the things here. And so the sparsity structure is something that we built something called the property DAG. And again, searching for property DAG and some collection of names will get you that paper, but it's a way of saying the sparsity has structure. And so you can bring back these classical ideas about active learning and experimental design and actually get them in practice. Now again, that's all wonderful, mathematically rigorous, a lot of fun. The people who did it had fun. But to actually then put this into an active drug discovery process where we have robots, people with their own careers and separate non-computational career objectives, we have drug discovery projects running at full speed. We were able to actually work with a lot of really great people, but I keep mentioning one person in particular, which is, there's a couple Wus on here, but Yan Wu, there she is, yeah, all right, is the head of antibody engineering at Genentech, and the two of us have worked together with this as a collaboration, a deeply embedded collaboration. And this allowed us to actually run this for 12 targets, for 4 or more iterations for each target, and show that for many different properties, so at the top I show sort of a distribution, and then the properties along this way, we have different seeds for different targets. And as we optimize and diversify those seeds, we stay in distribution for things like solubility, predicted immunogenicity, et cetera, et cetera. And here, so that we could show this to you, you were using the therapeutic antibody profile from the Dean Lab. We have our own version of these distributional metrics in-house as well. If we look at expression and yield, that Pareto front that I showed before, we see that as we go through iterations, we push that boundary out. And in that paper, we also show that for most of these targets, we got 100-fold improvements in affinity. We were able to mature almost all of the seeds for all the targets with a pretty cost-effective number of sequences. We can integrate this with libraries. There's all kinds of different sub-stories that fall off of those. Some of those are in the 782 appendix, page-long appendix of that paper. So, if you have insomnia, dig in. The other sorts of things that we're looking at are, again, how some of the most important advances in machine learning might not just change our bottom line or our speed, but they also might change our core ways of approaching the science of making good medicines. And I think we're very excited at Genentech about de novo hit finding and de novo design of antibodies. And again, there's lots of different things that we could talk about, how we integrate that with library design, how we integrate that with process automation. But I think the key thing is to just take a beat and realize that as we get these de novo hit finding, de novo protein design methods online, it's also going to give us a lot of opportunities to be a lot more rigorous in how we test structure-function hypotheses and a number of other things that underlie how drugs work. And so we can always optimize binders, but we can also identify where they bind. We can actually start to be much more rational about how we approach complex formats, which are increasingly important at Genentech, and actually have been a major part of our business for many years. And the road goes on and on. And so I think we think about optimizing binders, we think about timelines, but at Genentech, with a lot of the complex formats, we're thinking about having actual physical models of the complex, and the geometry of the therapy is really important to optimizing those things that have more than a handful of valences. So this is something we're really excited about. And again, this fits in the loop in the sense that that thing at the middle can generate antibody sequences all day long. And it can do it for all the formats that you have online at your company. And so somehow you've got to have a really high throughput, mathematically rigorous way of picking those things out. Now again, do you need optimal or do you need something that works? So again, if we're going for optimal, you need that. If you need something that works, then you can process optimize this in other ways, including ways of just minimizing the complexity. Another thing we've found with this is that using platforms that have more advanced machine learning to integrate workflows, suggest workflows, find data. Some of the stuff we heard about this morning, they also allow us to solve a really key problem, which is if we look at now, we're switching a little bit to our small molecule side, we have a lot of different things that people want to do. And here I'm listing things that are, let's say, core things that we might all agree a small molecule drug discovery platform would need. There's also a whole heavy tail of things that people like, different variants of different methods. And so, if you have a diverse group of scientists going after an objective like small molecule discovery, there might be hundreds of different tools they use. You can narrow it down, but ultimately what we find is that in order to dive into the different projects, each pipeline project has a different structure, function, hypothesis driving that discovery process. In the beginning, we had a lot of great things we developed, but we also had to deploy a lot of people to really get this into the hands of the people that were molecular leads on the pipeline projects. And the goal here is to have a platform that people can use instead of having to deploy your people in your computation, your highly skilled computational people into every project. Now, this is a lot to ask, again, because think of all the different mechanisms. If you look at the chart of the top 30 selling drugs last year, we've got peptides tied to soap. You got all kinds of funky things. You got venoms with medium molecules, small molecules, things bound to metals, and the metals are important. So there's so much structure function packed in to what we do as an industry that this system has to know about a lot of tools. It has to be super intelligent. It has to be the strength of system that we saw when we were discussing agentic systems. So, this is an example of just why what we're talking about isn't overblown. It's not more high tech than we need. We actually do need something that can navigate a lot of decision trees on the fly just to find the data in the first place. Okay. We're also working to develop our own models at Genentech, and I think this is something that we saw maybe different opinions in this morning. Some people talked about developing their own models. Some people were quite excited to just have open access models that were accessible in a nice platform like that, which the Bedrock platform provides. At Genentech, we want to make sure that our open access models can support the level of complexity that that platform I just showed you requires. And so we want to make sure that we can train it on our own data, and we also want to make sure that there aren't guardrails or data poisoning that's built for safety or built for good reasons, but also might confound a compliance or a safety issue down the line. So for those different reasons, we're actually building our own agentic systems with both open access models, but also with Roche trained models. And again, like we heard Dan say this morning, you often have different tasks that really strongly demand different types of models. And so we're training our own dedicated task-specific suite of models, and we're putting them together, both the larger models and the task-specific models, into these agentic frameworks. And what that looks like for us is that we actually have constellations of models. Some of the models are all-encompassing multimodal models, the larger Genie models, and these are some of the models that exceed the numbers that we've got here, but we find that for all kinds of deployment issues, for all kinds of HPC issues, we find that 70 billion is the sort of practical limit in order to make sure the inference times are low and that we can put these in the inner loop of different processes without blowing out the budget. So we've got our big models, multimodal models. We also have our models that are adapted to these agentic frameworks. So models that have deep experience with molecules and the different modalities. So in that first bucket, for example, is our universal molecular encoder. And then from there, we go into things that are more focused on reasoning, tool calling, and there's a lot of work going on with finding data and putting it into both the reasoning agent and molecule. So each of these models has its own, let's say, ability to find the data it was trained on. We hope to release these versions of models, so we will have essentially a version of some of these subsets of models. We're still working out the details of what we can and can't release. But for us, I think it's very important that we release as much of Genie as we're allowed to, because a lot of this was based on public knowledge. A lot of this is something where an open source community using our models will probably also find bugs, help us, and do all these other sorts of things that compelled IBM to make Linux its main operating system many years ago. And so we have similar ideals there, and we'll see what subset of our functionality we can release. But the legal team at Genentech is fantastic, and we're very optimistic that that'll be something this summer that we can share. When we put these things together, this is the type of, I just wanted to show one example of what this sort of looks like in practice. Agentic models can embrace the long tail. They can be super intelligent and know everything that's on the internet. But there's also a lot of cases where agentic models can optimize some of your inner loop, very common tasks. And so this is something where we have a question from a user about a set of seeds. So we have a set of seeds. We want to design 100 antibodies, perhaps for each of these seeds, that target a certain target, and they optimize these various parameters. Now again, is it a multi-format? Is it this? Is it that? Did you actually screw up and specify the wrong light chain? There's a lot that this thing has to check behind the scenes. It goes through its list, and to do that, it goes through both the list from a procedural perspective, but it also goes through different layers of the tech stack, as shown there, from the workflows to the tools to the actual interfaces. We lost a few boxes on the interface thing, but those are things like deposit back in a database, queue up, the order the antibodies interface, put them in our interface for molecular biologists to examine the sequences and double check that we didn't remove a cysteine that we shouldn't have, et cetera, et cetera. And so from the search and find the tools all the way through to the actual making the designs, we're very excited about this being an interface in addition to being a driver of innovation and a driver of essentially getting more out of a person's work hour. We also think this is removing a lot of questions and hard to maintain interfaces and other sorts of things. As you push the cutting edge, making interfaces to those tools is really tough. And doing that at a sort of enterprise scale is sometimes really, really tough. So this actually is a way of, just with a text-based interface, getting a whole bunch of access to these tools with a pretty general approach. So we're super excited about that. Well, this brings me to the end where I think it's good to take a step back and realize that my people can move a lot faster. And a lot of what we do relies on this underlying HPC infrastructure that is just absolutely fantastic, the AWS ecosystem, some of what we heard about today and lots of things that are down in the weeds that we won't get to. But the scale of all the compute behind what I've discussed is phenomenal. And big thanks to AWS for all the support on pretty much every facet of that. So thanks.